I'm not quite sure. So good day, everyone. So thank you for joining this TEIN summer workshop. And TEIN is a Trans-Eurasian Information Network, and we at the second phase, Dr. Tan and even Malaysian Society and Dr. Tan's colleagues, they kindly joined this effort. So according to the tradition of our the project, this year we extended our project to the virtual online one instead of the on-site here, Seoul National University, Korea. So we could just kind of expand our opportunity occasion to everyone. So thank you, Dr. Tan, and thank you. I'd like to appreciate the participation of the Malaysian Society. So this during this workshop, we might just they talk about the radiopharmaceutical chemistry, which is closely related with everyday routine of the Theranostics. So I'm going to just introduce Dr. Suh. And Dr. Suh is a co-organizer of this workshop. And also Professor Yoon Sang Lee is going to appear soon. Okay, thank you.
Hello everyone. So I think
it's okay. Let them start.
I think Dr. Tan will start the introduction for this workshop, and please welcome Dr. Tan and Dr. Lian.
Hello, hi everyone. So this is Miss Ng Yilian. So she will be our moderator in Malaysia. So I will pass the mic to Miss Ng. Okay.
Okay. Hello. Thank you for organizing this event for us. We thank you to Seoul National University. Okay, we have a number of participants that have logged in. We have specialists, doctors, we have pharmacists, we have radiochemists, we have physicists, okay, in our Malaysia side, and we're all very grateful that you're organizing this and we can learn more from you. Okay. 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 So we will start at 1:10. Okay. So in a few more minutes, okay, we let everyone come and settle in first.
So Mr. Suh, who is the first presenter? Are you the first presenter?
Yeah, Dr. Suh. Yeah, Dr. Suh is going to run the first session. 서미석 선생님 시작해야 되는데.
네. Yes, I will start the introduction. So I think it's just an introduction, so I can start now, and I think everyone can join through my introduction. I think it will take about 8 to 10 minutes. So I will start the introduction. So, okay. Okay, so hello everyone. Welcome to the 2020 TEIN workshop for Malaysia. So this time, our subject for this workshop is regarding radiopharmaceutical chemistry from clinical needs Theranostics. As there is an emerging interest in the field of Theranostics, we would like to focus on the aspect from the radiochemist. So actually, we have extracted keywords of nuclear medicine from the Journal of Nuclear Medicine 10 years ago, which was published 10 years ago, from 2010 to 2011. Interesting, as you have expected, the words that come out from these where the frequent words are placed in this box. And you can see that, as you expected, PET and FDG had a high frequency from these journals. However, after 8 years, we again extracted the keywords from the Journal of Nuclear Medicine from 2018 and 2018. And interestingly, although the PET still are the most frequent word, we can see that there are some words that are newly coming out, such as PSMA or therapy or treatment. So if you see what are the nuclear keywords that are increased or which has a high frequency in the journal, so are PSMA or PRRT, dotatate or immune, so which are actually dedicated to the therapeutic aspect of nuclear medicine. So what is Theranostics is a compound word of therapy and diagnostic. And as you can easily imagine, it is doing therapy and diagnostic in the same time. As the medicines has changed to precision medicine is now emerging, there has been a need to select a patient who will get the response to the treatment. The advantage of nuclear medicine is that we can see the patient who will get the response to the treatment. And since the nuclear medicine has its characteristics of a ligand and which is labeled with radiopharmaceutical. But when we change the radiopharmaceuticals, we can change this drug into an imaging method, or we can also use it as a therapeutic purpose. So when we image the patient, we can see who will respond to the treatment, and then we can treat the patient. But this concept is not a recent one. We already have been used this as a radioiodine therapy. But now there are more options that we can use for this diagnostic purposes, such as for neuroendocrine tumor, we have an option for lutetium dotatate therapies. So as you can see, we can image by diagnostic gallium dotatate whether this patient will have response to the PRRT or not. So we can select the patient. The market size of Theranostic actually has been increasing. And as you can see from the start of radium therapy for bone metastasis, there has been an increase of the size of radiotheranostics. And after the approval of lutetium dotatate, Lutathera, now there are more options of drug coming in the near futures. So actually, back in 2009, as you can see in this news title, that this radiotheranostics was known to be just unusual radiological treatment. However, now big pharmaceuticals are having interest for this radiotheranostics. And recently, Novartis has bought the AAA and also the endocyte, which products the PSMA. And for AAA, it almost cost $3.9 billion, which actually reflects the hype of interest in this field. So what is the process of this development of radiotheranostics is that there are many steps that are needed. So first, we need a basic research, and we can find out some number of candidate compounds. And then we have to see we should validate. And then after validation of this drug, we can proceed on to the preclinical research. And with this information from the preclinical researches, we can move on to the clinical trials. And after these trials, finally, the drugs can be approved. But as you can see, the the number of compounds that finally gets approved significantly decreases in every steps. And after the approval of drug, this should be introduced into the clinical guideline to be served as a regular practice. So as you can see, the Lutathera, actually the lutetium dotatate therapy has been introduced to the clinical guideline of ENET. And so now it has been a option for the therapy of neuroendocrine tumor patients. So what kind of works does a radiochemist will have a role in this process? So actually, in the earlier part of the drug development, radiochemist has a lot of role for developing and drug discovery, and also in the field of preclinical researches. However, there are also an important role after the clinical application of this drug. So actually, this is a gallium dotatate PET-CT. As you can see, this patient had a high numbers of liver metastasis and pancreatic lesion. So this patient underwent a lutetium dotatate PRRT therapy. However, as you can see in the right side of the image, you can imagine what kind of image this is. Actually, it seems like a bone scan image. However, unfortunately, this was a scan, post lutetium dotatate scan. So what happened? So then what should we do? So that kind of quality control is important in the clinical application of these radiodiagnostic agents. So this workshop will actually contains a sessions for the radiodiagnostic development, from the development to the clinical application. But also it contains a session for the quality control and the role of radiochemist in the quality control. So I welcome all to this 2020 TEIN workshop, and I expect you will enjoy the sessions. And I would like to ask if you have any comments in the middle of the lecture, you can just write down on the chat. And then after the lecture, we can answer to that comments. And also if you have comments after the lectures or questions, you can just freely ask us. So thank you. And we will start with Professor Yoon-Sang Lee. So please welcome Professor Yoon-Sang Lee.
Hi, nice to meet you. I'm Yoon-Sang Lee at the chemistry team, and I'm a radiochemist and also organic chemist. So mainly earlier period, I did chemical synthesis also I can do, and sometimes at that moment I did radiolabeling by myself. And firstly I finished the master course of chemistry department, and then I joined for Professor Jae-min Jung, who is the immediate past president of the International Radiopharmaceuticals Society. And he asked me that firstly he wanted to make a myocardial imaging agent that can be labeled with the technetium 99M. And at that time, we have a very limited resources. I don't know the situation of the exact situation of Malaysia, but at that time we have only chemical hood and one rotary evaporator for organic synthesis. And our lab located in the public zone of the lab bench. And at that moment, we cannot do the animal spectrometry by ourselves. So we have to send that animal sample to the our main campus. So it would take more than 1 month to get the one animal spectrum. So at the time we have a very long time to make a compound and also to confirm the chemical structure of that myocardial imaging agent. But after 10 years, we got almost all equipment for radiosynthesis, even an organic synthesis material also. So I think you better start first. So I think even though you don't have any organic chemist in your department, you can ask to make that chemical for radiolabeling. You can ask to other department in your hospital or institute. So start is very important. Starting is important for everyone. So how can we start? We have to select the target for that radio pharmaceutical. So and then after that, we can make chemicals for radiolabeling. So you can design that chemical structure of that compound and then organic synthesis. And after that, you can label it with the proper radionuclides. Sometimes if you want to make a diagnostic radiopharmaceutical, you can label diagnostic radionuclide. And sometimes if you want therapeutic radiopharmaceutical, then you have to attach the therapeutic radionuclide. And after that, you can do the in vitro experiment with that radio-labeled radiopharmaceutical, and then next you can do the animal experiment for in vivo or ex vivo biodistribution. And if you have an animal scanner, then you can get the image. For us, we don't have an animal imaging facility till the year of 2007, 6 or 7. Before that, getting that animal PET machine, we have to do the in vivo or ex vivo biodistribution study, and then sooner or later, if we got the animal imaging, then we can make a full series of paper like that. And after that, after finishing the animal experiment, you can confirm the efficacy of that radiopharmaceutical. Then you can do the GLP grade GLP grade toxicity study with the proper institute, and then after that, whole result will be filed up, and then you can make a investigational new drug application for in case of Korea FDA or FDA government body. And after finishing the clinical phase 1, 2, 3, and then you also once again file up all the results, and then you can submit the new drug application. And after approval of the government body, you can use that Radipharmaceutical for patient in the clinic. So which target you have to select? For example, for a brain receptor imaging agent, we have to consider the that can be passed through the blood-brain barrier. So that is rate with hydrophilicity or hydrophobicity, and also molecular weight is very important. And if you want to make a cancer targeting radiopharmaceutical that can target surface membrane antigen or protein, and you have to select any kinds of 3D structures such as antibody or nucleotide or any kinds of nanoparticles that can be a candidate for radiopharmaceutical. So to select the target, that is a very, if that has a good future, then that is very good. But you have to focus, you have to search all the references or the, you can use your knowledge to search the target. And then finally, if you select the target, then you can pick the kinds of protein structure of the target, and then you can make a peptide or antibody or small molecules. And after that, you have to consider the biological process of that target and radiopharmaceutical affinity. So you have to consider the half-life of radionuclide, because of that biological process. Sometimes it will take a longer time than the radionuclide. So half-life is very important for the selection of the radionuclide. And also for therapeutic or diagnostic purpose, you can select the beta minus or alpha for therapeutics, and gamma or beta plus will be the good diagnostic radionuclide. And then radionuclide can be attached to the that targeting molecule. Then you have to consider the chemistry for radiolabeling, that the feasibility of the radiolabeling to the precursor. And also we have to consider the molecular weight and lipophilicity and charge that can affect the biodistribution. Firstly, molecular size is very important for biodistribution. As you can see, the small molecule that can pass through the blood-brain barrier, and that maximum limit will be the 1,000 Dalton for small molecule. Sometimes you can use a large molecule such as a protein or antibody or even nanoparticle that can normally above 100 nanometer that can be captured by the liver corpuscle. And so if you want to prolong the blood circulation, you have to select the less than 100 nanometer sized molecules. And sometimes we can use the colloidal type radiopharmaceutical that can be used for lymphatic images or sentinel node detection of the tumors. Here you can see the charge of the radiopharmaceutical. For a brain imaging agent, we have to select a neutral charged one because of that a charged compound cannot pass through the BBB. And also positively charged pharmaceutical that can be uptake by the myocardial tissue. So positively charged can be used for a myocardial imaging agent. And negatively charged compound can pass through the fastly pass through the kidney. So normally that kidney imaging agent has a negatively charged compound. And also hydrophilicity is very important for biodistribution. So hydrophilic compound cannot pass through the BBB and can fastly excreted through the kidney excretion. And hydrophobic compound can pass through the BBB, but highly hydrophobic, that is related with the log P or log D value that can capture our hepatocyte with the protein binding characteristics. So after injection of that hydrophobic compound, sometimes that can capture in our blood system, then that protein carry out to the hepatocyte and it can degrade with the hepatocyte system. So you have to select the proper hydrophobicity or log P value for proper target application. So if you want to make a brain imaging agent, that log P value is that range, optimal range is 0.5 to the 2, around 2. So that hydrophobicity also very important for the biodistribution of the radiopharmaceutical. And next one is the how can we synthesize that precursor of the radiopharmaceutical? You can normally we use the organic synthesis method and also inorganic synthesis method that can be possessed at the metallic component. And sometimes we can separate the natural product of the that chemical component. And sometimes we can use the biochemical or biological synthesis method. So as I mentioned earlier, if you don't have a proper organic chemist in your department or institute, then you can ask to make other colleague in the chemistry department, or you can order to make that compound in the proper company. Or as I know that several Chinese lab, they can make order-based chemical synthesis. So I think it's I think that is somewhat expensive, but if you don't have any source for commercially available of that radiopharmaceutical, then I think you can think about that order-based synthesis. And as you can see here, several types of radioisotope you can select for diagnostic purpose: F18, C11, technetium, gallium, and other imaging radionuclide. And for therapeutic purpose, iodine-131 and rhenium, yttrium, lutetium, and actinium-225, and others. And then you can use adequate chemistry method for labeling. Then, and after the radio labeling, you have to do the purification step. Normally, a hydrophobic compound that can be separated by the C18 cartridge method. And sometimes we can use the ion exchange column chromatography. And sometimes we can use the high pressure liquid chromatography system. And after getting that compound, we have to do the quality control of that radiopharmaceutical. So after the quality control, we can get information of the radiolabeling efficiency and radiochemical purity, and sometimes specific activity for, especially for brain imaging agent. And pH is very important for injection because of the pH, safe pH range is around 4 to 8. And also chemical purity, I think after the short column purification, we think that there is no chemical component, but we have to confirm the chemical purity also after the radiolabeling. Here you can see there are only 3 methods for radiolabeling of the chemistry. So you can see here F18 and C11, they use SN2 reaction of the nucleophilic addition reaction. And other part are the coordination chemistry for metallic radionuclide. And finally, iodine-related compound, halogen compound, such as iodine series of iodine and also bromine compound radionuclide can be labeled with electrophilic substitution. And after the labeling, you have to do the in vitro experiment. And firstly, you can do the stability study with that labeling, that solution of the radio labeling, and also you can put that radiopharmaceutical into the serum, human serum, and then you can get the stability data up to the 4 times of half-life of that radionuclide. And sometimes we have to check the charge of that radiopharmaceutical with the electrophoresis or ion exchange column chromatography method. As I mentioned, that the octanol distribution can give the log P value of that radiopharmaceutical that is related with the lipophilicity of that radiopharmaceutical. And if you want to know that the affinity to the target protein of with our newly developed radiopharmaceutical, we have to do the binding assay with the proper protein or proper cell line. And then you can get the saturation binding and also specific binding, and then you can get the KD or KI value of the radiopharmaceutical. So you can confirm that is potent or not with the KI or KD values. Normally, for imaging or therapeutic radiopharmaceutical, that range is around up to 100 nanometer molar and down to the picomolar concentration range. And in vitro metabolism, you can do with serum or grounded tissue that radiopharmaceutical mixed with the serum or tissue, grounded tissue, and then you can check the TLC or HPLC, that analytical HPLC, then you can get the information of the metabolites of that radiopharmaceutical. And after the in vitro experiment, you can do the animal experiment with the proper animal model. Firstly, in case of newly developed radiopharmaceutical, you have to do the whole biodistribution of the radiopharmaceutical. Then with the normal animal. And you simply injected the radiopharmaceutical into the mice, and then you can get each organs, and then you can put that organ into the gamma counter. Then you can get the whole biodistribution of each organ, and then you can get the figure of the biodistribution or table of the biodistribution. And sometimes we can get information of the pharmacokinetics or pharmacodynamics data with the biodistribution or images. And sometimes we can do the autoradiography that can we can see the where the radiopharmaceutical is located or localized in the living animal. And after injection of that radiopharmaceutical, you can freeze that animal. Then we can cut the brain or other organ or whole body. Then you can see the map of the our radiopharmaceutical location. And if you have animal scanner, then you can image with the animal PET or SPECT or CT or MAD. And earlier we used the human gamma camera for animal experiment. Sometimes we can use the pinhole collimator for animal experiment. And then from that imaging data, we can calculate the binding potential of that radiopharmaceutical to the target. And also we can confirm the efficacy of that radiopharmaceutical with the proper animal model or tumor model like that. And after that, we can get information with the in vivo metabolism using the after injection of radiopharmaceutical. You can get the blood sample and the urine sample or feces sample. You can inject into the HPLC system, then you can get the information of the metabolites of that radiopharmaceutical. And after that, we can do the preclinical test for toxicity. So I think that is a non-GLP toxicity, and the other one is GLP toxicity. So I think maybe Min Suh will introduce the whole process of the our application of the that IND process for with that preclinical test also. And we have a lab bench and also our own lab in our department. So that is dedicated for chemical synthesis and also radiolabeling. So we have a cyclotron one, or we have a animal spectrometer and mass spectrometer and HPLC and module for radiosynthesis and TLC scanner and animal facility for imaging. So this is the end of my presentation. I think after Min Suh's presentation, we will have a discussion session. Thank you very much.
Okay, so if you have any comment for Professor Yoon-Sang Lee, you can write down on the chat, then we will answer it right after this. So what I would like to share is about our process of first clinical trial, radiotheranostics clinical trial in Korea, which was approved in 2020. So as previously mentioned, there is an emerging interest in the radiotheranostics. However, in Korea, the application of clinical PRRT was limited until now. So actually, the neuroendocrine tumor is growing in Korea. However, the number of patients is still near 2,500. And it is actually in the definition of rare disease. And as a rare disease, there is actually no registry system for neuroendocrine tumor in Korea. And there are many therapeutic options now available worldwide for the PRRT, and which has been recently introduced was the lutetium dotatate Lutathera, which showed a great effectiveness for the treatment of neuroendocrine tumor. And for this kind of effectiveness, FDA have approved this drug in 2018. However, and in Korea, however, there are limited options right now. Actually, the nuclear medicine itself has a large volume. We do a lot of clinical studies and diagnostic imaging in Korea. And there are many international trainees coming to Korea. However, our patients in Korea, to get therapy, radiotherapy, they go abroad to Malaysia, China, or Australia, Singapore, and even to Europe to get this radionuclide therapy. So then didn't we do anything to get this clinical application of lutetium dotatate? But we actually started the attempt to apply this technique from 2010. So we got a project. However, because we joining to the PIGS, because of necessity of additional GMP facility for the radiopharmaceutical, actually this project halted, and this clinical application actually has been delayed. But since, as you have seen earlier, that there is an increasing number of neuroendocrine tumor patients in Korea, and there has been a continuous public demand for this therapy. So we actually have continuously had consultation with the Korean FDA, and we finally got the IND approval. So we have now, in 2020, we have now started the first in Korea clinical trial for PRRT. So what is the process to get the IND approval in Korea? I would like to actually share this part. So there are, firstly, Professor Yoon-Sang Lee has told us what kind of testings we need before moving on to the clinical trials. And to move on to the clinical trials, there are many contents that are needed to be provided to the Korean FDA or even the FDA. So for the animal preclinical studies, it includes exploratory studies such as pharmacokinetics, toxicology and safety, pharmacology and efficacies. And it also needs a GLP studies, which is dedicated to the safety characteristics such as toxicologies. And with these kind of contents are ready, then we can move on to the phase 1 clinical trial. So these are actually, I'm sorry this is in Korea, but these are some contents that are needed to provide when we apply for the IND application. So there is mainly 2 parts that are needed. So one part is about the product itself, the drug substance and drug product. It includes the characteristics of the drug. And the second part, and also this about the product, it contains about the stabilities And for the next part is about the preclinical experiments. And this preclinical experiment actually is also divided into two main parts, which is toxicity and efficacy. So efficacy can be tested in our lab-based facilities by using like tumor models or biodistribution study or imaging. However, toxicity studies should be done in the GLP facility in Korea. But actually there, as a characteristics of radiopharmaceutical, most radio theranostic agents has one part of ligand for the targeting and one part of radiopharmaceutical for therapeutic effect. But actually, these toxicity tests are dedicated to see a chemical toxicity. So that it means that it wants to see the toxicity of the ligand itself. However, in the nuclear medicine, we use a very tiny, very few amount of ligand. So actually, Korea FDA and also the FDA does not require all kinds of toxicity data when applying to the IND. So genetic or reproductive toxicity or carcinogenicity of these ligands can be, is not necessary. So only in now, single dose or repeat doses are required. And so actually the toxicology for radiotherapy has a characteristic that it should actually test a toxicity for the ligand and also the toxicity, radiation-induced toxicity. However, as the radiation-induced toxicity is well known from the studies, we can, if we have a data of the animal biodistribution and dosimetry study, actually we can speculate the toxicity of this animal. So these can be actually done, the radiation-induced toxicity can be done using this biodistribution and dosimetry data. And actually the Lutathera also have provided the biodistribution and dosimetry data. And for the ligand-induced toxicity, I have already told that there is a need just for a single dose toxicity, single dose or repeat dose. However, there is some concern what kind of product we should use. When we use a hot form to characterize a ligand-induced toxicity, actually, if we multiply the dose for application, it will not induce a ligand-induced toxicity, but there will be more effect from the radiation itself. So actually the FDA or Korean FDA recommends us to do this ligand-induced toxicity using the cold form of the radiopharmaceutical. For instance, if we use a lutetium-177, there is a cold form of lutetium-175, then we can label it with that and then test the ligand-induced toxicities. So these cold pharmaceuticals are used, and as you can see from the Lutathera therapy, they have provided a single dose toxicity test using labeling with lutetium-175. But still, there are concerns of doing biodistribution study and using cold radiopharmaceutical for this ligand-induced toxicity or radiation toxicity. Because if you think of a ARPA particle, and some ARPA particle doesn't even have a cold radioisotope, which can be used as a substitute. And also because it cannot be imaged, they need a surrogate marker for the biodistribution study. However, actually in now Korea, actually there is some concerns about these alpha particles, but there is no still yet consensus about how to evaluate the toxicities of alpha particles. So these are some two characteristics of toxicity when we apply to the IND approval of clinical study. And also when you use a radiation toxicity, we have to evaluate the late radiation toxicities. And actually these late radiation toxicities are warranted when patients have a long life expectancy. But like patients who get PRRT, such as PSMA and neuroendocrine tumor patients, actually has quite a long life expectancy, which warrants this late radiation toxicity. And they recommend to do this dose should be at least twice the maximum planned human dose or radiation tolerance for the critical organs. So actually in the Lutathera, as you can see, repeat dose was done using a cold radioisotope. However, they used a double amount of 15 millicurie to see some late radiation-induced toxicity or to confirm the nephrotoxicity of this Lutathera. So again, these are some contents that are needed to be submitted to the IND. But what we had what took a long time for consultation with the Korean FDA was that this kind of documents that are needed, this kind of regulation was actually set up at 2019. So it takes a long time for us to have a conversation with the Korean FDA how to deal with this radiotheranostics. So now there is a guideline for theranostic radiopharmaceuticals for radiopharmaceuticals. And what I want to lastly emphasize that to have a breakthrough, you must participate in the process. Actually, we actively participated in the regulation process, and that was, but it took a long time, but finally we have breakthrough to start our first in Korea clinical trial. So thank you very much. And if you have any comments, you can ask freely.
It's free now. He might be in the middle of doing a report, but let me answer on his behalf. He answer.
Can speak, right?
Can.
But I ask you to answer a question.
Can I ask you? Okay, sorry about that. Okay, so in Malaysia, for PRRT therapy, we have two drugs. We have lutetium dotatate and we have yttrium-90 dotatate. We don't use Lutathera. We are using labeling. We will label the lutetium and the dotatate. And for yttrium dotatate, there is no available labeled product. We also perform the labeling. Then we perform the QC, and then we give the dose to the patient. So PRRT, we have two options. We are trying to further a new option of actinium-225, but not, for now we're still in the process. Okay. Other than PRRT, in Malaysia, we also have lutetium-177 PSMA. So yeah, these are the three options we have for radionuclide therapy, other than iodine.
Is there any planning for the PSMA? As you mentioned already. Okay, thank you.
Actinium, yes.
Actinium,
yeah, 225 PSMA. We are interested in actinium-225 PSMA. Many Korean patients also ask us about actinium-225.
Okay, I see. Thank you.
So is it only a requirement for this kind of practice? Do you need a IRB process, or is there some regulation from the like FDA regulation for use of radiotheranostics in Malaysia?
In Malaysia, we are a bit different than in Korea and in America. We not necessarily use products with FDA registered. Sometimes we use products from Europe. So if they have gone through phase 3 clinical trials, they are safe to use in patients, we would choose to use them as well. So in Malaysia, we have different regulation for using drugs. Okay, we have our own local regulations for products registered in Malaysia. If they are not registered in Malaysia, then we would apply. We call it as an import permit to import those drugs from overseas and use it in our patients.
Okay.
Thank you very much for sharing. So then, is there any other comments or questions for this session? So if there isn't, we can end the session right now, and we can move on after some break. So we will have a, I think, 10-minute break, so we can